Hello class, this is your lecture on stems. Let's get started. The outline is as follows. We'll look at the external form of a woody twig. We'll look at stem origin and development. We'll look at stem tissue patterns. We'll look at herbaceous versus woody stems, and then for the woody stems compare dicots to conifers. We'll contrast all of these with monocot stems. We'll look at a short list of specialized stems, and we'll say a few words about wood and its uses. So first, some review. We have seen these terms previously in the semester, but we want to go over these one more time in the context of our stems lecture. When we look at a woody twig, we can say that the woody twig consists of a central axis with attached leaves, and we know that the leaves attach at the nodes. This leaf arrangement can be said to be alternate, opposite, or whorled, and the space between the nodes is called the internode. Finally, the leaf has a flattened blade and is usually attached to the twig by a petiole. Now, at that little angle where a leaf petiole attaches to the stem is where we would expect to find an axillary bud. And that axillary bud is typically going to be a region where lots of cells are dividing. Therefore, it can grow into more stem or even a flowering stem. At the tip of a growing plant, we would expect to find a terminal bud. In some plants, we also find stipules, and these are paired appendages found at the base of a leaf. They often remain on the plant throughout the leaf lifespan, and these are really characteristic of certain plant groups only. For example, we often see stipules in members of the rose family. One of the most challenging things for a botanist is to identify a tree species based upon the bare twigs after the leaves have dropped. As you can imagine, the leaves and their characteristics can be very helpful in identifying a tree. Now, whenever a tree drops its leaves annually, typically in the autumn when it gets cold and the days get shorter, those trees and shrubs are referred to as deciduous species. And after those petioles fall off, they leave behind some scars. These leaf scars can be examined closely, and inside of a leaf scar, we might find a bundle scar that showed where the vascular tissue used to connect the leaf to the stem. In this diagram, it shows us a micrograph of a dividing leaf and stem tip. And remember, apical meristems are those special meristems that are found at these tips of actively growing regions. These contribute directly to stem length, which is therefore primary growth, and often apical meristems go dormant over the winter and then start dividing rapidly in the spring. These apical meristems, if they're damaged, means they will no longer grow at that part of the plant. So therefore, they're often protected by bud scales and even the leaf primordia. The plant would rather sacrifice its baby leaves rather than a meristem because it can always grow more leaves later on. These leaf primordia are actually tiny embryonic leaves that eventually develop into mature leaves. Recall that apical meristem cells form 3 primary meristems, and this is where cells are all dividing by mitosis. The first is the protoderm. This region gives rise to the epidermis. The next is the procambium. This produces primary xylem and primary phloem. And finally, the ground meristem. This produces pith and cortex, both composed of parenchyma cells. Later on, we'll figure out how to distinguish the locations of these pith and cortex regions. Both leaf primordia and bud primordia develop into mature leaves and buds. And as we're examining these different parts of a stem, it can help us look at the development when we examine these parts. So first off, we have these traces. So traces branch off from cylinders of xylem and phloem and eventually enter a leaf or a bud. And so these traces are very important for a biologist that wants to see the structure and development of different plant groups. Now, as these traces, which are essentially strands of xylem and phloem, go into a stem, it often leaves behind a gap, and that's just based upon the architecture of moving from a stem to a petiole. But in the advanced plants, they fill in that gap with parenchyma, and we're going to talk about those plant groups later on in the semester. Now, as these stems are growing in girth, which is to say lateral growth, we'll have narrow bands of cells that form between the primary xylem and the primary phloem, and this may become a vascular cambium in some species. For example, any species with woody tissue are going to have to develop a vascular cambium. The cells produced by the vascular cambium become components of secondary xylem towards the center of the stem and secondary phloem towards the outside surface of the stem. Now, in many plants, the cork cambium is going to also work alongside of the vascular cambium, and the cork cambium is ultimately going to produce the bark. The bark of that tree is composed mostly of cork cells that are impregnated with waxy suberin, as well as phyloderm cells that are produced on the other side of our cork cambium. The primary purpose of producing bark is to reduce water loss and protect the stem from injury. But because those cork cells are filled up with waxy suberin, it doesn't allow the bark to breathe very easily. So in many species, they will produce lenticels, which are loose groups of parenchyma cells that allow for more efficient exchange of gases. Now, as we're examining the internal architecture of a stem, a biologist might describe the steles, and notice the spelling is S-T-E-L-E-S. This is the central part of the vascular cylinder, and it refers to the arrangement, or rather the spatial arrangement, of the vascular bundles. This includes the primary xylem, the primary phloem, and the pith if present. Now, the stele arrangement often allows us to classify different types of plant groups. For the purposes of this class, we don't have to memorize the specific types of stele arrangements. We just need to know the general definition. Next up, let's start comparing and contrasting the differences between different plant groups as this applies to their stems. First, a quick review. Dicots are flowering plants that develop from seeds having 2 seed leaves, and monocots are flowering plants that develop from seeds with a single seed leaf. And these seed leaves are what we call the cotyledons. Now, in some species, and this is really about the herbaceous dicots that we're talking about now, we will refer to them as annuals. That is to say, these are plants that die after growing from seed to adult in one single growing season. These plants are usually green herbaceous plants, but there are exceptions to this. Now, most monocots are annuals, but many dicots are also annuals, and throughout the entire plant, most of the tissues are largely primary, which is to say they only experience primary growth in these organisms. Now, herbaceous dicots in their stems tend to have discrete vascular bundles that are arranged in a cylinder. And so when we examine these under a microscope, we'll see these herbaceous plants with these bundles that look like they're just in a ring inside of the epidermis. Now, in some cases, these plants will develop a vascular cambium between the primary xylem and the primary phloem. So they might have a certain degree of secondary growth, but it's typically only in the bundle. It's not throughout the entire stem like we would see in a woody plant. Remember, we're talking about herbaceous plants in this example. The other main way to distinguish an herbaceous dicot stem is to notice that the interior part of our stem is completely filled in with pith. Pith is a region that mostly is made up of parenchyma tissue. Now, what about those dicots that do have woody tissue? Well, wood is essentially just secondary xylem, so it's going to be the result of cell division on that vascular cambium. And also, we have to consider the court cambium if the tree is developing a nice thick layer of bark. There are some differences in woody tissues. For example, some plants, such as African mahogany, have a vascular cambium and court cambium that is active all year. They grow in kind of a drier tropical forest where the conditions are just good year-round. So because of that, the wood has a distinct appearance. It's typically ungrained and uniform, which is why it's prized for many human uses, including furniture. Now, in most trees, there are growing seasons. There's a spring, summer, autumn, and winter. And if the wood is produced seasonally, then we would say in the spring, the tree produces spring wood. In spring wood, there's usually a lot more rain and moisture, so the tree produces large vessel elements. After the spring wood forms, usually a summer drought comes on. It gets hotter and drier, so the tree produces fewer smaller vessel elements in proportion to tracheids and fibers. And again, this is in the context of a dicot stem. Now, in conifer stems, that includes things like pine trees and redwoods. Those are going to produce woody stems as well, but it's very different from a dicot woody stem. So in conifers, vessels and fibers are absent. Instead, their wood is made pretty much exclusively from tracheids. And again, they do produce spring wood and summer wood, but there's no vessels, only tracheids in that typical conifer woody tissue. Now, 1 year's growth of xylem is referred to as an annual ring, and this applies to any woody stem, whether it's a dicot or a conifer. And a biologist can count the growth rings to figure out exactly how old that tree is. Now, the vascular cambium, remember, is largely producing this woody tissue, and it produces more secondary xylem than phloem. This is why we see these nice packed-in growth rings, because the vascular cambium is working so hard to produce this woody tissue. Vascular ray tissue usually is composed of parenchyma cells, and these parenchyma cells aren't going to be dead at functional maturity like the xylem is. Instead, this parenchyma simply works by having water diffuse from cell to cell for the lateral conduction of nutrients and water. We find the vascular rays in both the xylem and phloem regions. Xylem ray simply means it's part of the ray within the xylem, and the phloem ray simply means it's part of the ray through the phloem. Now, if we look at a woody stem and we see a crack that's going from the center to the outside, that's probably evidence of where a xylem ray used to be, because parenchyma cells don't have a secondary cell wall. So when they dry out, they leave those cracks behind. In this diagram, it shows us a woody stem, specifically a young dicot stem, that shows pith in the center, 2 growth rings in the secondary xylem, a vascular cambium, and then the secondary phloem regions finally giving way to the bark. When we examine a stem, we can look at it based upon its surface. And this will be a bit challenging in this course to figure out which surface is which, but by studying the diagrams in the textbook, I hope that you will be able to describe these surfaces. We have the transverse surface, the tangential surface, and the radial surface, and we'll be describing these in much more detail in just a little bit. Our next term is a tyloses. So tyloses are protrusions of adjacent parenchyma cells into the conducting cells of the xylem. So after the xylem stops functioning, we often want to fill it in with special parenchyma cells that we refer to as tyloses. This prevents infection from things like bacteria or fungus. And over time, it eventually darkens as it fills in with resins, gums, and tannins, and then we refer to it as the heartwood. The heartwood, therefore, is the older, darker wood in the center of our stem, and the sapwood is the lighter, still functioning xylem that's closest to the vascular cambium. Other terms include hardwood versus softwood. Hardwood is the wood of a dicot tree. Softwood is the wood of a conifer. Recall that conifers don't have fibers or vessels, so that's why we refer to them as softwood species. Another term is a resin canal. We find these mostly in the conifers, such as pine trees, but we can also find them in some hardwood species as well. And these resin canals are tube-like canals scattered throughout the xylem and even sometimes the phloem. And they secrete resins that serve primarily as antifreeze and to prevent herbivory. Now, bark is very important in many woody species, and the bark refers to the region of tissues outside of the vascular cambium, including the secondary phloem. And mature bark may consist of alternating layers of both crushed phloem and cork cells. Here's an example of the oldest single-stemmed clonal tree on our planet. It's only 16 feet tall, and it's found in the Swedish tundra. This Norway spruce might not seem that impressive, but don't judge a book by its cover. This tree is nearly 10,000 years old. Now, it's not the oldest tree on the planet per se, but it is the oldest single-stemmed clonal tree, meaning that while the trunk may have died off here and there, the same roots have endured this brutal tundra climate for millennia. Now, in tissues, sometimes we find things that are called lacticifers, and these lacticifers are ducts that are found mostly in the phloem, and they secrete latex. Some examples of common latex products include natural rubber and chewing gum and morphine from the opium poppy latex. Now, monocots are different from dicots and the conifer in their stems. In a monocot stem, they have neither a vascular cambium nor a cork cambium, and therefore they produce no true woody tissues. Instead, they have primary xylem and phloem in discrete vascular bundles that are scattered throughout the stem. Also, because these bundles are scattered, we say they have no distinct pith or cortex regions in their stems as well. Now, in a typical monocot vascular bundle, we'll see 2 large vessels and several small vessels, as well as a bundle of phloem. There's also typically air space, and this is where xylem might have used to have been, but the xylem cells stretched and collapsed during development. Finally, there's often a vascular bundle that gets surrounded by a sheath of sclerenchyma cells, giving protection. We often refer to this as the bundle sheath. In this diagram, it shows us how those vascular bundles are scattered throughout a monocot stem, and then we zoom in on a monocot vascular bundle, which almost looks like a little monkey face. We can see the eyes, which is the vessel elements, and the mouth, which is actually an airspace. On the forehead of the monkey is the phloem bundle. Now, some monocots look like trees. Think about a palm tree, for example, but it does not have real wood. A monocot, even if it has some degree of secondary thickening, instead has a different type of meristem. It has a secondary meristem that simply produces parenchyma to the outside and secondary vascular bundles to the inside. In the next part of our lecture, we're going to learn a short list of specialized stems. The first is a rhizome. These are horizontal stems that grow below ground. An example of a rhizome is tropical ginger. Next is a stolon. These are horizontal stems that generally grow along the surface. The common houseplant called spider plant produces stolons. Another example of a plant that produces a stolon is the strawberry. Next is a stem tuber. Things such as white potatoes have internodes at the tips of stolons that swell up after they accumulate starch. A bulb is considered a specialized stem. It's a large bud surrounded by numerous fleshy leaves with a small stem at the lower end. An onion is an example of a bulb. A corm looks like a bulb, but it's composed almost entirely of stem tissue. The plant called taro is a corm. Often people call it taro root because it grows underground, but now you know taro is actually a specialized stem. Cladophylls are unique. They are flattened, leaf-like stems. Things such as horse-tongue lily and cactus produce these cladophylls. Often, these cladophylls have leaves that are reduced to very small structures. For example, in the cactus, the leaves are reduced to pointy spines. Spines, therefore, are modified leaves. Next up, we've got thorns versus tendrils. And if we're talking about thorns and tendrils in the context of a specialized stem, then they must be produced from the axillary buds only. An example includes a passion flower and a grape, which produces stem tendrils. We're going to learn later on in the semester there are examples of leaf tendrils as well. It's all about where they originate. And if they originate from an axillary bud, then we know it's a specialized stem. Here's a quick comparison. In a leaf tendril, we see these commonly in members of the pea family, and these leaf tendrils are typically modified leaflets from a compound leaf. In the last part of this lecture, we talk about wood and its uses. Now, in a living tree, 50% of the wood weight comes from water content. The dry weight, therefore, is composed of 60 to 75% cellulose and about 15 to 25% lignin. When we're looking at wood, density and durability are very important characteristics for commercial products. Density refers to the weight per unit of volume, and durability refers to the ability to withstand decay by organisms. When we look at wood, we might see knots, and a knot is actually a portion of a side branch or a dormant bud that is included in the wood of the stem or a larger branch. These are often concentrated in the older parts of the log towards the center. All right, well, in review, we've learned all things stems now. I hope that you email me if you have any questions, and thank you very much for listening.